This is a production of Cornell University.
Thanks, Charlie and Anna, for the intro. Happy to be talking about some of my work looking at pleiotropy within maize. And so to start us off, I think it's helpful to define what pleiotropy is. And at its simplest form, pleiotropy is a single locus that affects multiple traits. And it can happen through a bunch of different mechanisms, of which you have a single causal variant that's a star, and you have a single causal variant that impacts two different traits, or two causal variants within the same gene that then affect two different traits. There's also mediated or vertical pleiotropy, where that single causal variant impacts one trait, which then subsequently impacts another. And then probably most commonly is spurious pleiotropy, where you have a region of really strong LD, making it hard to ascertain which causal variant is impacting your two traits of interest. And so within human disease, pleiotropy is a really common feature, with up to 44% of the genes in the human GWAS catalog showing pleiotropy. And of those pleiotropic sites, they've been associated with higher and wider gene expression across more tissues. They've been enriched in open chromatin regions, and then also shown some DNA conservation. And so within maize, there's a few examples of like cloned or verified pleiotropy out there. And of these are teosinte branched 1 that impacts inflorescence length, lateral branching, and branch length, along with VGT1 that impacts plant height, leaf number, and flowering time. And so there's this assumption in the early 2010s that with more genotypes and with more phenotypes and association mapping, that we'd be able to find more pleiotropy. And so this is one of the first stabs at that, looking at pleiotropy in inflorescence traits and flowering time, and found that essentially there were no shared loci between all three traits. However, there was some sharing between tassel and ear traits, or things sharing the same like developmental units or pathways. There's also some pleiotropy between tassel and flowering traits. And most of the pleiotropy that's present between flowering leaf has to do with maize making sure it has the right number of leaves before it initiates flowering time. And so more recently, a paper came out a few weeks ago looking at pleiotropy across 162 different maize traits. And very, very few of those SNPs genome-wide actually do show pleiotropy. And of those are between traits that we would kind of already expect to share loci, like flowering time with leaf architecture or flowering time and inflorescence architecture traits. And very rarely do we see sharing between like flowering time and root genes, which is unexpected. And so it kind of leads us to this question of why is there so much more pleiotropy in human disease compared to maize? And so a couple of key limitations that might be biasing how much pleiotropy that we're seeing is that we're mostly looking for pleiotropy within maize mutants versus standing natural variation populations. And a lot of these pleiotropic profiles assessed through large effect mutations may really differ from these small effect larger populations. Also, GWAS models in maize have historically had low resolution. And so trying to determine which is the causal SNP from many candidates still remains elusive. And also results may vary by the model that you use, the types of covariates, the types of traits that you select, significance thresholds, and pleiotropy statistics. And so with all that together, we wanted to ask how explainable is pleiotropy in maize through a bunch of different metrics. The first thing that we need to know is there any variability in pleiotropy whatsoever? And if so, can we potentially bring in the things that were important in human genetic studies, like gene expression, DNA accessibility, DNA conservation, and then also add in statistically interfering variables that we know impact GWAS studies, like the amount of LD and the number of SNPs that we use to map those traits. And so all together, this kind of builds a really cool model that we can then test to see how pleiotropy is impacted and controlled within maize. And so in order to do that, we use two existing mapping populations, the Nested Association Mapping Panel, or NAM, and the Goodman Panel. And with our hope with all the phenotypes that are already available in these populations, is we would get phenotypic saturation in order to uncover the genome-wide distributions of pleiotropic loci across the central dogma of traits. And so we're starting around with 32 million traits between our two populations, RNA expression values from 7 diverse tissues, metabolite features, and then field features like flowering time, plant height, and inflorescence architecture. And so this is what our overall pipeline looks like, where we have our different populations, our different trait types, and then also the number of traits that we're mapping with. We then perform GWAS within those populations, and then aggregate those results by interval. And so, for example, here we have 74,000 intervals genome-wide, and our unique trait SNPs associations are those different shapes. And so for interval A, we have 2 unique traits mapping to it, so we mark it as 2. For interval B, we have 1 unique trait mapping to it, so we count it as 1. We do that for the rest of the intervals genome-wide. And so then we get a count of the number of unique traits that map to each interval genome-wide. And so in order to get something to compare these counts with, what we do is we permute all of our phenotypes anywhere from 5 to 10 times, run them through this same exact pipeline in order to get a null distribution of what no pleiotropy looks like whatsoever. And so I will note before I start showing some plots that our pleiotropy is a single genic or intergenic interval that can affect multiple traits. And we don't distinguish between what type of pleiotropy we're looking for. So it's any one of these mechanisms that could potentially be happening. And so before I show you the plot, I want to describe what you're going to see. And so along the x-axis is going to be the bin number of traits that are mapped to that particular interval. And so it could be anywhere from 0 to a few thousand traits. And along the y-axis is the permuted minus original proportion of intervals that fall in that trait number category. That's kind of confusing. So anything along the horizontal line means that there's no pleiotropy. And then anything above means also it's noise, probably not pleiotropy at all compared to the permuted data. And anything below the line means that there's some pleiotropy that's promising to look into. So with that, this is just showing the results for the NAM field data. And this bump right here is probably a region of strong LD and probably just noise altogether. But there is this little promising bump that might show some pleiotropy that has anywhere from like 30 to 90 traits mapping to it. I will say that the number of intervals showing this trend is on the fraction of a percent genome-wide, so it's a very, very rare occurrence. And also I don't collapse on unique traits, so I can have multiple versions of flowering time represented in this plot across different environments. But still, there's something interesting here. And so looking across populations and different trait types, these distributions change wildly. And so for the Goodman field traits, which is in the top right-hand side, there's significantly less pleiotropy, but there's still some there for a very small number of intervals and traits. And then for the Goodman metabolites and expression, there's very, very little pleiotropy whatsoever, and it's on the fraction of a percentage of intervals genome-wide that show this trend. And so now that we know that there is variability in pleiotropy, we can go forward and build models that try to explain this. And so what we do is take all of those biological features from before and then run a model with our pleiotropy statistic. And so this is our random forest results, where along the x-axis is relative importance, and along the y-axis is all of the different features that we use to build our model. And so everything in blue are statistical features and gray is biological. And so the most important features overall are all of our statistical things, like the amount of LD, the number of SNPs that we use, and the interval size. And of very low importance are all of our biological terms, like expression, like conservation, and so on. And if we look across populations, we see pretty much the same trend of there's very little importance of all of our biological terms compared to our statistical terms. And so that kind of leads us to say that pleiotropy is not really associated with many biological features within maize. And so I will note, even though I don't have time to show it today, we did create an adjusted pleiotropy statistic, removing all of the variance from those statistical terms, and still found the same relationship of gene expression, accessibility, and conservation isn't associated with pleiotropy compared to the permuted data. And so to kind of conclude things, we hypothesized that pleiotropy is not common in maize. And of the few pleiotropic loci that do exist, they can come from sites with varying amounts of gene expression, chromatin accessibility, and sequence conservation. Also, pleiotropy in crop plants is highly constrained by evolution. And this kind of makes sense because the independence of traits is essential if you're going to adapt new varieties to new environments. And that's especially true out in the wild, and also within breeding programs. Independence is nice to have. And also results still may vary by the type of the species that you use, the types of traits that you look at, the marker resolution and saturation, and of course, not having a robust null to actually compare all of your results to, which we have here. And with that, I'd like to say thanks. I am funded off of a USDA NIFA fellowship along with some other grants within the lab. And I'd like to say thanks to a bunch of the folks in my lab and happy to take questions. There's a question online. Great talk. Biologically, we can anticipate that many genes are pleiotropic through regulating the development of plant phytomers. Can you developmentally, biologically interpret your results and observations that are obtained from statistical associations? Developmentally, well, I guess to kind of answer your question, we do some like go enrichment to look at the types of genes that might be, I guess, highly pleiotropic, but I'm not sure if that answers your question. If you could follow up with me, that would be great. John Luke.
Great talk, yes. You started out with the question of why is maize so different from humans, but you didn't give an answer. What's the answer?
I think it's selection. Different types of selection and the bias in human genetics to study diseases. And we're not necessarily looking at those same types of traits, and they might have different genetic architectures control, or they do have different genetic architectures. And I think, yeah, with diseases, we're biased to studying things that are wrong, not necessarily things like more quantitative like other plant traits. Yeah. Yeah, Anna.
Thank you for talking about this. I was wondering how the selection of your phenotypes might affect whether you can find pleiotropy or not. Because you might then have all the metabolite data, but then look into very detail of specific metabolite things that you'll find pleiotropy that you don't find between like metabolites and flowering signs.
Yeah, I think it is really biased, and I think that's been kind of like addressed in some papers, in that if you're just looking at components of the same trait or like ratios of metabolites that are all highly related, they probably will show more pleiotropy because they have, yeah, a shared regulatory pathway. And so, yeah, trait selection really does matter in something like this, where it looks more prevalent, but across many, many more things, it's not necessarily a common phenomenon. Yeah. Okay, Rebecca, what's your question?
Maybe it's related to Jean-Luc's, but yeah, thanks also for a great talk. And I'm wondering, I mean, surely there's other data about humans that's more analogous to the types of data about a plant that you're mentioning. So it seems funny to me to compare human disease versus corn anything. And so I'm wondering if what does human anything get you in the way of pleiotropy? And then what if we look at plant, corn disease as a corn pathologist, of course, I can't help wondering. And I think there's a lot of pleiotropy about other traits that affect diseases via that indirect type of pleiotropy, presumably. So just curious if has anybody looked at it other than disease? And what is the analogous question, really, to make those two comparisons actually more analogous? How would you set it up? Because I'm unsatisfied by disease versus other, as a comparison.
Yeah. I should mention that it's not only in human disease where pleiotropy is common. There are other traits that they've looked at and seen a little bit more pleiotropy than normal. I think disease is where it's most pronounced and also gets the most funding and papers, which is a little biased. But yeah, I'm not sure what an equivalent version of this would look like in maize. Maybe disease, yes.
Because we look at how to
for a larger, the field traits, I think,
influence the ability of fungi to colonize that air and produce toxins, for example. But I think anyway.
Do I still have time for another question? Thanks, Rebecca. Okay, yes, John Luke.
If you could re-explain your robust null, that would be helpful for me.
Sure. So what we did was essentially permuted all of our phenotypes anywhere from 5 to 10 times. And then we removed the variability from the PCs that still might be holding population structure, and then use those like PC adjusted phenotypes as our new phenotypes, and then map those. So yeah, does that answer your question? Okay. All right.
Alrighty, hello everyone. My name is Elise Tomaszewski, and today I'm going to have a discussion about the approaches that I've taken to try and understand the regulation of fruit abscission in Physalis grisea. So Physalis grisea can be characterized as a Solanaceae crop that has a sprawling growth habit and an indeterminate growth habit as well, meaning that the plant continues to produce fruit and mature them throughout the entirety of its life cycle. So these fruit are about the size of a grape tomato. They have the flavor profile of a cross between a mango and a pineapple, so pretty sweet and also tart. And Physalis grisea is part of a genus of around 100 different species, but it is closely related to pepper and then also related to tomato as well. So it is part of the Solanaceae family. So to get everyone oriented on the Physalis floral anatomy, I'd like everyone to look on the right here, which is part of the Physalis flower. We have the petals here, the stigma and styles, and a structure called the calyx or sepal, which surrounds the flower. And then we also have a pedicel, which is kind of like a stalk that connects the flower to the plant. Once the flower is then pollinated, a globe-like fruit will then be produced, and surrounded by this calyx or a husk is what it's normally called. Something that's interesting is that a lot of people think it's the flower that is surrounding the fruit, but in fact, it is this calyx. And on this bottom left here, you can see that the petals are actually pushed down towards the end of the calyx. But the part that I'll be talking about most here is the pedicel, and this is where the abscission zone forms and where the fruit detaches from the plant. So what is abscission? There's three main types of abscission that I'll be talking about briefly in this part, and one that I'll be talking about for the rest of my talk. So the first one here is leaf abscission. This is characterized mostly, you can see around Ithaca in the fall when leaves are falling off of deciduous trees. You also have flower abscission, where unpollinated apple blossoms will abort from the plant due to limited starch and sugar allocation. And then fruit abscission is where I am mostly interested in. This is beneficial for the plant because it'll allow for seed dispersal. However, while most abscission is beneficial for the plant, agronomically, this is not the case. And the fact that when you're dropping fruit, it is unsellable and you cannot have a profit then as a farmer. Also, you'll have increased labor for trying to clean up this fruit or increased harvest time, losing out on profit again. So I'm using Physalis grisea twofold. One is a model to understand how fruit abscission is regulated both spatially and temporally. And I'm trying to figure out what is specific about fruit abscission, where, when you're dropping your fruit, you aren't also dropping all of your leaves at the same time. That's not beneficial for the plant at all. On the other hand, I'm trying to reduce abscission in Physalis to make it a more agronomically viable crop for farmers. I'd also like to point out here that Physalis grisea drops its fruit while other plants do not. And what I mean by this is that tomato will have this abscission zone that forms on the pedicel as well, but it's not raining fruit, kind of like how Physalis does. So to get everyone oriented exactly on what abscission is, here's a hypothesized model that I've created to outline the 4 different steps of abscission. The first here, and what I will be showing, is the petiole, which is outlined from a hypothesized tomato plant. On the left, you can imagine that the fruit would be attached, and then on the right, the petiole would be attached to the plant. And I use petiole and pedicel interchangeably, just so you know. Within the petiole, a few layers of cells called the abscission zone cells, which are characterized as small cytoplasmically dense cells, form. And then after that, these cells have to become competent to external regulation, such as plant hormones like ethylene or ABA, which then induce cell separation by dissolving the middle lamella as well as degrading the cell walls. And then after that, the plant then produces a form of lignin shown on the left side of the fruit side of the petiole here to act as a mechanical barrier to kind of help set up the plant where this break should happen. And then the abscission zone cells on the plant side of the petiole then confer some sort of epidermal-like qualities, and they produce a cuticle to act as a protective layer. So I'll be talking today about a couple of genes that I'm targeting specifically for steps 1 and 4 to try and reduce abscission in Physalis. So to do that, I am using a reverse genetics approach to knock out different genes, looking at the literature of what has been done before in closely related species like tomato, as well as Arabidopsis and lychee. So the 3 genes of interest that I'll be talking about today are J, macrocalyx, and IDA, which is downstream of abscission zone development. But what I'd like to point out here is that J and macrocalyx are Madsbach's transcription factors, which in the literature have been found to interact and form protein complexes with other Madsbach's transcription factors. And in this case, they are developing the abscission zone cells or inducing abscission zone development. So I will be targeting these to hopefully knock them out and then reduce any abscission zone formation. So the first gene that I will be talking about today is J, which is the homologue of jointless in tomato. So J is in Physalis, jointless is in tomato. It is a MADS box transcription factor, which regulates floral organ development. Jointless 2, which is the homologue of jointless in tomato, there's a lot of homologue going on here, creates this joint over when it's knocked out, does not have a joint. So, however, this homologue does not exist in Physalis grisea. That would be my first choice as it's used a lot in the commercial variants, but that does not exist in Physalis. So I'm going to jointless to see if I can knock out an abscission zone, which this diagram shows here on the left, wild type pedicel where a joint or a knuckle is formed where the fruit will detach from the plant. And then on the right, when the jointless gene is knocked out, you do not have any joint formation. So hopefully that is going to happen with my Physalis plants. So I've been able to transform using a CRISPR construct that I've designed to knock out the J gene in Physalis. I've been able to take them out of culture, and I now have T0 plants that are growing in the greenhouse where I'll be able to collect fruit, self them to hopefully T3 or T4 generations, and look for any differences in abscission zone cell formation, as well as any other effects of knocking out this gene. The next gene I'll be talking about today is MPF3, which is the homologue of macrocalyx in tomato. Again, it is a MADS box transcription factor, and in tomato has been thought to interact with J. So I'm hoping to knock out multiple parts of this protein complex that is theorized. In this case, in tomato, when you knock out this gene, you have both large calyces, which are shown in the diagram on the top right, compared to the wild type, but it also interacts with J. So you aren't forming an abscission zone. So on the left here, you have an abscission zone and the wild type, and then on the right, this line of demarcation is not there. So there's an incomplete abscission zone being formed. So I'm also hoping to knock this out. And I do have T3 lines now and find that the homozygous mutants do not drop their fruit as much as the wild type compared on the right here. I would like to notice or point out though that there is an abscission of the fruit later on, but it does seem delayed. I'd also like to point out here that the size of the pedicels between the wild type and the mutant, shown the wild type on the top and the mutant pedicels on the bottom, have a very clear size distinction. So the mutants being much larger, but they do have this abscission zone. You can sort of see it as a white line in the middle of the pedicel. So I'm interested to keep looking at how this is affected on the cellular level. And then the last gene that I'll be talking about today is inflorescence deficient in abscission. It's been characterized in Arabidopsis and lychee for floral abscission, as well as fruit abscission in lychee. But it is a peptide ligand that binds to leucine-rich receptors that induces a cascade to induce upregulation of cell separation genes. So that's step 3, 4 that I'm talking about with my hypothesis of the abscission zone model. So the first row on the top here is wild type Arabidopsis flowers, and the bottom is what I'd like everyone to focus on as well with the knockout of IDA. We can see here that around stage 9 in the wild type, it's dropping all of its floral organs, where when you knock out this gene in Arabidopsis, the petals remain attached to the plant. This doesn't mean that there's not an abscission zone, it just means that the cell separation has not been able to occur. So I'm hoping to knock this out, and I've been able to create another CRISPR construct. And I now have these in culture, and I'm hopefully going to have them progress along. So I'm very excited to give this talk a year from now, and hopefully I'll have more going on. I have a lot more projects that I'm currently doing, such as an RNA-seq project looking at a developmental series to understand differentially expressed genes within the abscission zone to hopefully target them. I'm also looking more at microscopy to have a quantifiable way to show that there's a difference between the abscission zone cells within my mutants and my wild type, as well as using force measurements as a way to quantify any differences between the mutants and the wild type. So the detachment force is what I'm looking at here. And then lastly, one of the other ones I'm trying to do is then create double and triple mutants. So crossing my MPF3 and my J mutants as they're part of MADS box transcription factor pathways, and see what this does also to the abscission zone development. So I'd like to acknowledge all of my committee members, Joyce Van Eck, Jim Giovanoni, and Adrian Roeder, as well as especially the Van Eck Lab, specifically Savannah Dale, Carrie Swartwood, and Nathan Ream, who is a former postdoc, for all of their continued help. And I would be happy to take questions and also give recipes if you'd like. Yes.
Fantastic talk, Elise.
Thank you.
When you mentioned that you ruled out jointless too, can you go into a little more detail on how you move it out. Things like assembly quality in your species can affect, maybe it is presently raining.
Right. No, that's a good point. So I blasted the genes. That's how I've done it before. And I've been able to find jointless or MPF3. And I didn't see anything that had high homology between jointless 2 and any gene within Physalis. Our reference genome is quite new. But I do think it would, I believe within the literature, there is no jointless 2 within a Arabidopsis either. So this might be a specific thing to tomato to have this jointless 2 and interacting in a protein complex. Yes.
I'm wondering, so when you harvest the Physalis, when I do not have the Abscission,
Mhm.
do you expect the fruit brings off the plant? So that
Yeah.
there is some balance between not wanting to have an Abscission zone, but not having to have the fruit drop.
Yes.
You don't want to pull the whole plant apart when you take the fruit.
Right. And yeah, I couldn't get it into my talk, but I'm really looking at a dosage effect in a way. So having kind of this intermediate phenotype of an abscission zone. Right now, I'm using it as a model. I'm trying to figure out what these genes do at first, but I would expect them to break off. So you have this abscission zone on the pedicel, but you also have an abscission zone where the fruit connects to the calyx. Let's see if I can quickly go back. Okay, so you also have an abscission zone by the fruit with the calyx. So I would expect there to be this abscission zone where the pedicel and the calyx connects. So it's kind of like tomato, where when you break it off in jointless tomatoes, the pedicel and the sepals remain on the plant, but you just get the fruit, which is advantageous then. So you're not having a bunch of poked fruit due to the pedicel. So that's what I would expect. Cool. Yes.
So you do take the fruit and the I see that in tomatoes sometimes that the fruit kind of breaks. Do you see that in
I haven't yet. And some of the mutants I've seen some splitting on the bottom of the fruit itself, but that's pretty uncommon in wild type. And all the mutants that I've had, I haven't seen that yet. So no, but I'll keep an eye out for it too. Okay, thank you.
Thanks, Charlie. Hi, everyone. Today, I'm going to talk about one of my projects examining effects of the Manihot glaziovii introgression in cassava. So cassava breeders in the 1930s were facing a disease epidemic of cassava mosaic disease. And to try to combat this, they were breeding for disease resistance by making crosses with cassava's wild relative, Manihot glaziovii. And progeny from some of those crosses became important founders in modern breeding germplasm. So the legacy of that is that there are 2 large introgression segments from Manihot glaziovii on chromosomes 1 and 4 in cassava. And so you can see in green and blue, the introgression segments on the last 10 megabases of chromosome 1 and the central 20 megabase pairs of chromosome 4 increase in frequency as cycles of selection proceed. And so the introgression is becoming an increasingly important part of the population structure. And so previous work in our lab by Martin and Wolf et al. Looked at how the chromosome 1 introgression affects root number and dry matter percentage, which it affects yield as well as the textural properties of cassava roots. And so what's interesting is that the chromosome 1 introgression doesn't seem to be affecting variability for disease resistance, but it does impact these important yield traits. However, they also found that there's suppressed recombination in the introgression region. So the introgression tends to be inherited as a big block, and that makes it difficult to break unfavorable linkages in this region. And it also limits the resolution for us to tell what's going on with the QTL of this region. And in addition, we find that there are almost no homozygous introgressed lines in advanced trials. So we see homozygous introgressed lines in early stage trials, but they get selected against very quickly. And Martin hypothesized that this might be due to deleterious mutations that have accumulated in the introgression region. And so breeders have to maintain the introgression in the heterozygous state, which is somewhat costly because whenever you cross 2 lines that are heterozygous for the introgression, a quarter of your progeny will be homozygous introgressed and then automatically are unlikely to be advanced. So I'm interested in following up on this work by looking into what are the negative effects of homozygosity in the introgression. In particular, are they localized to particular segments, or are deleterious alleles spread throughout the introgression region? And then also with increased resolution, can we narrow in on the yield QTL and identify which segments contain this yield QTL and see if we can select only the segments that contain the yield QTL while purging the deleterious load that's carried by the rest of the introgression. So to generate high resolution for this QTL analysis, we started by making crosses to generate recombinations in the introgression. And we then screened the progeny with CAS markers, which was a way to look at just the introgression genotypes of many individuals. And then we evaluated these individuals for vigor, which I hypothesized might be an effect of homozygosity in the introgression, that they would have poor fitness for vigor. And then we use these genotypes to select a subset to advance to replicated trials where we can evaluate yield. And finally, I'm working on evaluating associations between particular introgression segments and these yield and vigor traits. So this is the seedling nursery that came from those crosses. And so this was growing at the International Institute of Tropical Agriculture in Ibadan, Nigeria. And the crosses were made and planted by a team led by Dr. Rabi and Smith Ikfan at IATA. And I got to visit this field over the summer, and so the crosses were made between 41 parents that were heterozygous for the introgression. So there are 259 families. And while I was there, I evaluated vigor both in plant height and stem diameter, which are two components of what makes a plant look vigorous to a breeder. And I also collected leaf samples so that we could screen this population for their introgression genotypes. And so to screen many individuals in a cost-effective way, we used a panel of 10 CAS markers. So CAS markers have a competitive primers that bind to a biallelic SNP. And so for this panel, I chose glaziovii specific SNPs that I identified from a panel of 118 accessions from cassava HapMap2 that either had the introgression or lacked it. And identified 10 of these SNPs across the introgression spaced about 1 meg base pair apart. And we validated the SNPs using the parents, which were already genotyped, as well as a set of wild glaziovii trees and hybrids between esculenta and glaziovii. And you can see from the diamonds that those wild and hybrid individuals clustered where we would expect them to in the homozygous and heterozygous genotype classes. So with those 10 markers, we can get a granular view of the introgression genotype for the individuals in the seedling nursery on the y-axis across the 10 CAS markers on chromosome 1 in the introgression region, spaced about 1 megabase pair apart. And so, for example, you can see one individual here lacks the introgression on this half in the pink, and it's homozygous for the introgression on this half in dark blue. And so we were able to observe recombinations between each of the marker pairs. And so between 11 and 358 recombinations between each pair. But you'll notice that there are fewer homozygous introgressed individuals in dark blue than there are homozygous non-introgressed individuals in pink. And so to test if that was significant, we did a chi-square test to test if the genotype distribution was significantly different from the 1 to 2 to 1 ratio that you'd expect from making crosses with heterozygous parents. And we saw that it was significant. There was fewer homozygous glaziovii lines than you'd expect. So this could indicate that there is some kind of seedling or seed viability or early survival issue with the homozygous lines. And this is just for CAS marker 1 that I'm showing, but we saw the same pattern that was significant across all 10 markers. So next to look at if homozygosity in the introgression affects plant vigor. So here I'm showing stem diameter as a measure of vigor and broken out by the genotypes across the 10 CAS markers. And I saw that the heterozygous introgressed lines in light blue had slightly higher stem diameter than the homozygous lines. And to test if that was significant, I used a mixed linear model for stem diameter with the additive and dominant effect for each marker, counting for family and field position with random effects. And I saw that several markers had significant effects for stem diameter. And so the effect sizes for those markers for stem diameter is significant. You can see for markers 3 and 6 through 10, the dominance effect is significant. And so that explains that the heterozygote has a slightly higher stem diameter than the homozygotes, but none of the additive effects are significant. So it doesn't explain why the homozygote, it doesn't explain that the homozygote glaziovii line is any worse necessarily than the homozygote esculenta lines. So returning to this question, why are homozygote lines, why are introgressed homozygotes excluded from advanced trials? It looks like glaziovii alleles have statistically significant but quite small effects on vigor. So going back to this, you can see that all the effect sizes are between 0.25 and 0.5 millimeters. So it's quite a small difference in stem diameter. So it seems like that could be contributing a small amount, but it's definitely not fully explaining why we almost never see homozygous introgressed individuals in later stages. So I'm thinking that the segregation distortion we observed, as well as possible impacts on yield, may be more important factors in explaining what's going on with the negative effects of homozygosity and the introgression. And so to evaluate yield next, we needed to select 300 individuals from the population of about 3,000 that would represent the genetic diversity present in the introgression and seedling nursery, so that we can replicate those individuals and get better measurements for their yield in clonal trials. And so to pick the 300 from the 3,000, I used an optimal design algorithm with the opt-Fedorov package in R. And so this algorithm picks a subset of individuals that captures the maximum amount of information about the marker effects and the parent effects. And so these are the genotypes from the 300 selected clones. And you can see that they're enriched for the recombinant individuals. So it's providing more information about the independent effects of the markers. And so next, this clonal evaluation trial is growing now at IATA, and I will return in August to for harvest season to look at the yield traits from this trial. And we're also doing whole genome DART-seq genotypes for these individuals. So I'll be able to look at rest of genome effect versus introgression marker effects, rather than just having a parent effect. So hopefully that will give us more information about isolating the effect of the introgression versus background. And I also want to follow up on the segregation distortion that I observed and see if there are structural variants in the introgression region that might be explaining that. With that, I'd like to thank my committee members, the Buckler Lab, for sharing cassava hat map data with me, as well as our collaborators at IATA who have set up the trials and helped me in the field and made it possible for me to study this population. Thank you. Yeah, the question was, is germination rate scored? And it is not, and that's something that we thought about doing at the beginning, is having a germination experiment where we try to genotype the seeds that do not germinate. Yeah, we didn't proceed with that because it is somewhat difficult to get seeds here, and it's quite labor intensive to do that and figure out how to extract DNA from the seeds that didn't germinate at IATA. And since I'm here for most of the year, it wasn't feasible for me to do that project there. But I think it is, we did try and genotype as early on in the season as possible to capture as many of the genotypes as we could.
But from what your visual observations, or what we could say failure to germinate, was it like 10% or 20% of the seed planted in that range?
I don't have an estimate on the percentage of seeds that didn't germinate, but I think in general, it's quite high for cassava for any specific cross, where I think it's maybe 50 to 75% of the seeds germinate. Yeah, in general. So, yeah. Yeah.
I have a question concerning the stem diameter phenotype rate. You talked about that it's very free to use. That's kind of like a medial. Has it been like actually shown that it is linked to yield?
Yeah, so stem diameter is not typically looked at in the breeding trials. What is scored for vigor is a 3, 5, 7 scale for vigor, which is somewhat relative to where 7 is the best in the field and 3 is the worst for vigor. So when I was there, I wanted to get a more quantitative measurement for vigor. And so I talked to breeders about what parameters they're actually looking at to figure out if something is a 7 or a 3. And something that came out of that was that thicker stems and taller plants contribute to being scored a 7 versus a 3. And so that's the traits that I decided to measure, and stem diameter is not typically measured. So I will be able to see, I have some early yield measurements from the seedling nursery. They're not that reliable because there's no replicates, but the stem diameter was correlated with yield in that trial, but I think we'll have to wait to see how the replicated trial looks between the correlation of stem diameter and yield to know if it's correlated with yield. But in general, the above ground plant biomass doesn't always correlate with the below ground yield. Thanks.
This has been a production of Cornell University, on the web at cornell.edu.